Hi, my name is Trevor and I'm a PhD student and visualization researcher at Harvard Medical School. Today I want to share several computational methods that I worked on during my research internship with Ozette Technologies for the effective exploration and comparison of single-cell embeddings, like the one that you see here in this background. At a high level, my talk today outlines a set of computational approaches that we've designed to first transform single-cell embeddings into high-resolution, aligned representations, and second, characterize these aligned embeddings to surface potential relative shifts in abundance between them. Our main goal is to guide the visual exploration of such embeddings and to help surface potentially interesting phenotypes faster. So what exactly do I mean when I say single-cell embeddings? A single-cell embedding is a lower dimensional representation of an often very highly dimensional single-cell data. You can imagine a single-cell dataset like a large table where every row corresponds to a unique cell, and every column represents a different characteristic or feature of that cell. An embedding is a 2D or 3D representation of this information that attempts to summarize these data and can serve as an important tool to understanding complex relationships. Visualizations based off these embeddings may be used as a tool to generate new hypotheses, annotate clusters of similar cells, and compare annotations. And here are some examples of embeddings showing a single-cell dataset from flow, mass cytometry, and CiteSeq. While these visualizations open opportunities for the exploration and discovery, they often introduce their own challenges for interpretability and comparison. First, it can be challenging to distinguish between general and specific cellular phenotypes. For example, are we interested in the broad cellular generic phenotypes, or do we want to be able to visually differentiate between more specific cell phenotypes as well? Second, we grapple with batch effects, or variations that stem from differences in the experimental conditions across these samples. When embedding multiple samples, how should we handle these batch effects? Are we interested in the sample or cell differences? And lastly, there's a challenge of sample comparison. Even with very high resolution and aligned embeddings, identifying meaningful differences between phenotypes can still be a formidable task. For example, here we have two aligned embeddings, the high resolution embeddings, one for healthy tissue and one for a tumor sample. They are colored by cell phenotype, and you can see that the layouts look very similar. Take a moment to look at the left and the right, and you can see if any phenotypes stand out to you. Some of the larger phenotypes, or those with bright colors, might pop out first, but are there differences between the two embeddings? Did anyone catch this purple phenotype that's missing in the tumor embedding entirely? So how do we go about surfacing these phenotypes for comparison when visually exploring these embeddings? We developed a suite of computational methods to address these challenges and produce aligned, annotated, and comparable embeddings from cytometry data. We also introduced methods for surfacing insightful differences in cell type composition and abundance. The first step of this process is the generation of well-resolved and aligned embeddings through data transformation, which leverages the annotations of each cell. This stage is accomplished by annotating cells based off of their marker expression levels using a novel machine learning method called FAUST. Unfortunately, I don't have time to dig into the details today, but I encourage you to read the original paper to learn more about FAUST. This method was also presented last year by Fritz Letches at this conference, as well as Evan Green. So, without going too deeply into the technical details, at a high level, FAUST annotates the modes of each marker with human interpretable label, like a negative or positive expression. And then using these FAUST annotations, we then apply a data transformation which consists of 3 steps: the removal of outliers, the normalization of expression ranges for each marker, and finally, the translation of positive and negative expression modes for each cell. The figure on the right depicts the stepwise applications of these methods to normalize CD4 T cells in a panel with CD3, CD4, and CD8. Applying this transformation is useful for embedding visualizations as it results in well-separated cell type clusters and subsequently allows for the identification of rare and complex cell types. The annotation transformation also eliminates batch effects and aligns our embedding spaces across its samples. For example, on the left we have an embedding of cells from healthy tissue, and on the right we have a different aligned embedding of tumor cells. Next, we develop methods to compare these aligned and annotated embeddings. The individual cells in both embeddings are colored by this shared cellular phenotype. At a glance, it can be very challenging to navigate these embeddings and discover what cell types might appear different. But we can color these embeddings based off of the metrics that we designed to surface cell types with potential shifts in abundance and composition between these samples. So here, our metric guides the visual exploration of these two embeddings, employing a bright orange or bright blue color to phenotypes which appear more or less pronounced in one embedding compared to the other. So how does this comparison work conceptually? We break down our comparison into 3 steps that we call confusion, neighborhood, and size. Confusion measures the data point intermixing in the latent space to identify commonly confused labels. Neighborhood looks beyond the confusion region and identifies which other cell types are nearby in the embedding. And then finally, abundance compares the relative sizes of the labels that are identified within the local neighborhood. Each step of this process yields a representation that we can compute separately for different embeddings with shared phenotypes, and then compare between these two embeddings. So for example, in this contrived example, we have the naive T cells are less confused, share a similar neighborhood, and have a decreased relative size compared to the top embedding. We realize our conceptual framework as a traversal of a Delaunay graph for the separate labels in the embedding. First, we construct the Delaunay graph using the Euclidean distances in the 2D embedding space. Once we have this graph in place, we proceed with the label-specific operations. For each label in our data, we conduct a breadth-first search for every point associated with that label. So for example, let's again focus on the yellow cells here in the middle. We start by traversing the graph to define what we call the confusion set. We start with this first point and look at all of its neighbors, which are 1 hop away. We then move to the next point and repeat. And we move to the next point and repeat again, and we repeat this for all points that share the same label. When this iteration is complete for a given label, the union of all visited points defines the candidate confusion set. We then apply a distance cutoff based off the average distance between the labels of the same type. And then the final confusion set thus includes only those additional points which maintain similar distances to labels of the same type. Following the confusion set, we then define what we call the neighborhood set. The neighborhood set is constructed by exploring further nodes from these within our confusion set. We again traverse the Delaunay graph, but this time visiting nodes which are more than 1 hop away and not already within our original confusion set. This process effectively divides our graph into 2 distinct regions for each label, the confusion region and the neighborhood region. Finally, we apply a connectivity-based adjustment that normalizes the neighborhood set into a connectivity score. Each neighboring label is then assigned a likelihood based off of the number of connections and average distances between these connections. Once we score these connections, we can then compare the similarities of these connectivities between 2 separate embeddings. Since our metrics are intended to guide the visual exploration of the embeddings, we conducted 2 large within-subject user studies in order to refine our implementation. These studies helped inform and validate our approach and ensure that it aligns with human perception. Unfortunately, I don't have time to talk today about these studies in depth, but grab me after the talk and I'm happy to chat in more detail. With that background out of the way, let's get into some examples. We implemented our comparison metric inside Jupyter Notebooks as an interactive widget, enabling the guided exploration and comparison of embeddings. The user interface for the widget is broken down into several components. First, users can select combinations of markers which define the resolution of phenotypes in the embedding used for the marker selection. Second, each embedding contains 2 linked views, a phenotype view where each cell is colored by its phenotype, and a metric view where each cell is colored by its metric. Here, we're displaying confusion. This layout is repeated for a second embedding so that we can interactively explore and compare the left and the right. In this initial view, we have a standard UMAP embedding and an annotation transformed UMAP embedding for mass cytometry dataset derived from melanoma patients. Here we're comparing a standard UMAP versus our annotation transformed UMAP for non-responder data from unstimulated pembrolizumab samples. These are the same exact data, it's just that the annotation transformation is applied to the embedding on the right. We expect our transformed embedding to contain less visual intermixing of cellular phenotypes. And indeed, selecting confusion set on the left, which appears to be bright and yellow, highlights several different confused clusters on the right. In contrast, when I select a small subset of unconfused cells in the annotation transformed embedding, we can see the small subset of cells are spread between two large clusters in the standard UMAP. This is not surprising because the standard UMAP embedding only resolves the broad cellular phenotypes based off a few markers. In fact, we can reduce the phenotype resolution by selecting only a single marker, here CD8, and show that both embeddings capture these broad cellular phenotypes. At this level of resolution, you can see that the embeddings are both relatively unconfused. However, as we add more markers, the cellular phenotypes become more detailed. And there is more confusion in the standard UMAP embedding. The annotation transformed UMAP embedding, however, remains unconfused across all label resolutions, including when all markers are selected for the visualization. Next, we can switch the metric view from confusion to display the neighborhood composition differences. Similar to before, we can select shared cell phenotypes to view in both embeddings, this time based off the cellular neighborhood similarity. Neighborhoods that are most dissimilar are assigned a brighter yellow color so they pop out. And again, similar to confusion, we can view the neighborhood metric at different label resolutions. We see that with just a single marker, the neighborhoods are quite similar. However, again, as we add more markers, the neighborhoods become more dissimilar. This denotes greater variability in the local neighborhood composition for each label as we add more markers and have more high-resolution phenotypes. Now, instead of comparing identical data from different embedding methods, like in the previous example, let's take a look at two transformed and aligned embeddings from our previous dataset, the non-responders on the left and the responders on the right. As both these embeddings have undergone annotation transformations and alignment, they demonstrate very low confusion and high neighborhood similarity. This provides us the opportunity to further examine potential shifts in abundance. For each phenotype with the metric view is now colored based off the relative shift of abundance compared to its neighborhood. This coloring enables intuitive visual interpretation. Positive shifts are represented in orange, while negative shifts are represented in blue. We can select a bright orange phenotype in the responder embedding, which denotes a phenotype in the responders with a positive shift compared to the non-responders, according to our metric. The goal of our tool is to guide the visual exploration and help surface interesting phenotypes faster. However, in order to confirm any actual differential abundance differences, we need to employ a robust statistical framework. Here we did this by opening up the same exact phenotype in the OZET platform. We can see a similar embedding with the phenotype selected, but also sample statistics that confirm that this phenotype is truly differentially abundant. We can repeat this process again interactively, letting the metric guide our exploration of these embeddings and inspecting cellular phenotypes which might be of interest. One of the challenges with traditional methods for identifying differentially abundant phenotypes is a lack of context to other related phenotypes. Embeddings provide us with a visual tool to pinpoint groups of related phenotypes that exhibit similar shifts according to our metric. For example, this group of cells in the bottom right, which display a positive shift. Our metric is telling us visually that there are differences between these groups. But we can again examine these phenotypes in a more statistically robust framework to verify that these phenotypes are actually differentially abundant, and in this case, again, they are. To summarize, our annotation data transformation refines single-cell embeddings, optimizing downstream comparison of high-resolution cell phenotypes. These embeddings are well-defined and aligned, which allow us to design metrics to characterize and compare similarly transformed embeddings. These metrics guide the visual exploration of such embeddings, enabling quick discovery of interesting phenotypes. However, the validation of differential abundance requires the application of appropriate statistical methods. Thank you for listening to my talk, and thank you to everyone that made this project possible, especially the OZET folks for providing this opportunity for me as a grad student, and I especially want to thank Fritz for his mentorship and collaboration in this work. If you want to learn more about our comparative embedding tool, please take a look at our GitHub repo and try it out. Thanks.